Okay, so thank you very much for the introduction, as I said. And what I'm going to do today then is just talk a little bit about what I do in the Centre for Natural Products Discovery, focusing on the pathways that I look at, particularly this pathway called Nrf2, which DDEM actually mentioned briefly, and I will also go into some detail. So I'm like in the middle of a phytochemical sandwich. I think DDEM did a lot of phytochemistry. Jose's coming after me is doing a lot of phytochemistry, and I'm like the biological filling of that phytochemical sandwich. So I think you probably all agree, and a lot of you guys are interested, well, most of you guys are interested in natural products, if not all of you guys. And I think you probably agree with me that natural products are quite unique in pharmacology. Natural products cover the complete spectrum of going from prevention, preventing disease right through to cure. Now, I am particularly interested in cancer. That's my background, and that's what I'm interested in. I'm interested in both aspects of cancer, both the prevention of cancer, and natural products are very good at preventing cancer. And also, I'm very interested in the cure of cancer. That's literally causing, using natural products to cause toxicity in cancer cells, causing that cancer cells to die. And natural products are great because they cover that whole spectrum. There's very little other chemotherapeutics or biological agents which actually cover that spectrum. Tamoxifen might be one if you think about cancer. Tamoxifen being both a preventative and a treatment, but no, natural products do tend to do both that jobs quite well. So within the CNPD, I'm actually I specialize in chemoprevention. Chemoprevention is this idea when we look at cancer. It's the fact that pharmacological, natural, synthetic products, and we tend to specialize in natural products, can prevent cancer from happening. So this idea that can prevent cancer from happening, and that's much better than actually treatment of cancer because it's cheaper, it's obviously less stressful for the individual. So we're trying to prevent cancer from occurring. We do that in two ways. We either prevent the formation of toxic species, which is one way of doing it, looking at antioxidants, or the other way would be trying to initiate cell defense mechanisms, to induce mechanisms within the cell itself so that the cancer, that DNA mutation does not occur in the first place, and therefore the cancer does not begin to be initiated. So we're interested primarily in initiating these cell defense mechanisms with regards to cancer. Now, interestingly, most of our work is actually focused on this Nrf2 pathway, and I'd just like to thank this diagram is actually was actually made up by Dr. Hiba Jasm, who is actually here today. Hiba made this up originally. I've just borrowed it, as they say, from one of our presentations, I think. But this goes into the actual detail of Nrf2 and how it works. So, chemoprevention, one of the mainstays of chemoprevention is activation of this pathway called the Nrf2 pathway. So Nrf2, let me get my pointer up here. So Nrf2 is this transcription factor here. This molecule here, it's a transcription factor which basically sits in the cytoplasm of the cell. Under normal situation, under normal circumstances, is degraded. Okay, so this molecule here, KEAP1, is a ubiquitin ligase. This is the ubiquitin ligase, and it ubiquitinates Nrf2, adds ubiquitin onto this molecule, and it's degraded. Okay, so it never enters the nucleus. However, under conditions of oxidative stress, what will happen is the association between KEAP1, this ubiquitin ligase, and this transcription factor Nrf2 is broken, and Nrf2 will move into the nucleus of the cell and join to this ARE. ARE stands for antioxidant response element. ARE stands for antioxidant response element, and that is found. It's a common sequence found in DNA. Nrf2 binds to that and fires off all these cell defense genes. So cell defense genes such as phase 2 enzymes, such as glutathione transferases, hemoxygenase 1, NADPH coenoxidase reductase, NQO1, as you can see down here. Now, it also produces glutathione. Lots of other, approximately 200 genes are induced by this transcription factor binding to this DNA. And these genes are generally all involved in the defense of the cell against harmful effects. Okay. Now, interestingly, we can actually stimulate Nrf2 using phytochemicals. Okay, we can stimulate Nrf2 using phytochemicals, and this is what we're primarily interested in. We're interested in stimulating this Nrf2 to fire off these cell defense genes using phytochemicals. Now, how does it relate to COVID-19? Well, COVID-19, interestingly, there is evidence this transcription factor, Nrf2, is related or has a role to play in preventing the effects of COVID-19 infection. So first of all, it's interesting to note that lung biopsies from COVID-19 patients, this transcription factor Nrf2 is actually suppressed in lung biopsies for COVID-19 patients. Now that suggests to me that the COVID-19 SARS-CoV2, given its official name, SARS-CoV2, is trying to inhibit Nrf2 to allow its progression within that tissue. So it's suppressed in lung biopsies, suggesting that it's a role to play against that infection. And secondly, we also note that Nrf2 activators such as dimethyl fumarate, DMF is dimethyl fumarate, is actually an FDA-approved drug for the treatment of multiple sclerosis. And actually it's thought to act through Nrf2 activation. Thus, this Nrf2 activating drug actually inhibits COVID-19 infection. Okay, so there's two good lines of evidence there that suggest that COVID-19, activation of COVID-19 does somehow prevent or fight against COVID-19 infection, SARS-CoV2 infection. And just to remind you guys, as Didem mentioned, it's actually not the COVID-19, the SARS-CoV2 virus that causes the effect. It's actually the response of the human body to that infection. Now, the response is a cytokine storm that causes a huge amount of interleukins, interferons, macrophage activation, and eventually results in this acute respiratory distress syndrome, ARDS, which results in damage to the alveolar cells of the lung, leading to fibrosis, and then you get problems in breathing. Just to focus a little bit on the biology of that, this is the actual lung space in the cells, and these are the lung space of the lungs, and these are the cells that are affected, these type 1 cells which cover most of the lungs. These are the type 1 cells cover most of the lungs, allowing gas exchange to occur. And if they are damaged by COVID-19, that cytokine storm, these other cells up here, these type 2 cells can actually replace these type 1 cells. These type 2 cells are stem cells, and they will replace these type 1 cells. Unfortunately, these type 2 cells are also damaged by the cytokine storm. So we've got a real combination of effects in the lungs with regards to cytokine storm, the activation of macrophages, production of reactive oxygen species, damaging these cell types. That eventually results in fibrosis, loss of respiratory capacity, and a poor outcome for the patient. So the idea would be then to use Nrf2 to dampen the cytokine storm. And there is good evidence to suggest that Nrf2 represses pro-inflammatory genes such as interleukin 6 and interleukin-1B, especially in murine macrophages. And therefore, there is good evidence to suggest that Nrf2 can be activated to dampen the cytokine storm, prevent that adverse consequence of infection due to COVID-19, SARS-CoV2. This is actually the life cycle of the COVID-19 virus. This is actually taken from a very good publication, Quadardo et al. 2020, published in Trends in Pharmacological Science. And you can see here, this is the life cycle of the COVID-19 virus, SARS-CoV2. Actually, it binds to this receptors, ACE2 receptors, and then taken into the cell where it's replicated and then generally packaged up back in a vesicle, which is then exocytosed out of the cell to keep spreading that virus, release of mature viral particles. And what's interesting is that the fact that Nrf2 is found to actually impede this viral cycle at several different points. We've got Nrf2, and you can see it up here, actually been found to inhibit or assumed to inhibit through various other researchers' work to inhibit that ACE2 receptor to stop actually COVID-19 from binding to that receptor to get actual entry into the cell. Secondly, we've got Nrf2 inhibiting the STING molecule. This STING molecule is involved in the production of interferons. So if Nrf2 inhibits STING, we'll get less interferon production, which is good for reducing the cytokine storm. And thirdly, we've got the actual physical act of viral infection of a cell activates this kinase here called PR kinase, this kinase here, which actually phosphorylates Nrf2, activates Nrf2. There's lots of different pathways, but mechanisms within the actual viral cycle of the SARS-CoV-2 where Nrf2 can actually be involved in inhibiting that viral response. So interestingly, if you can see down here as well, we also got involvement of another Nrf2 regulated gene. So heme oxygenase 1. Heme oxygenase 1 is an Nrf2 regulated gene. It's switched on by Nrf2. And interestingly, now Nrf2 activating heme oxygenase 1 has actually been shown in previous studies to inhibit lots of different viruses. So we've got influenza virus, HIV, the hepatitis viruses, even dengue virus and the Ebola virus. No activation of this Nrf2 regulated gene, heme oxygenase 1, has been shown to be antiviral with regards to these viruses. How that actually works is actually been elucidated. So if we look at Nrf2 activating heme oxygenase 1, this enzyme, heme oxygenase 1 actually degrades heme into carbon monoxide, iron, and biliverdin. A bit like bilirubin, but it's actually biliverdin. Okay, so carbon monoxide, iron, and biliverdin. And this carbon monoxide actually inhibits NADPH oxidases. That prevents reactive oxygen species formation, and therefore we get the inhibition of that damage associated with hydroxy radicals, superoxide, hydrogen peroxide.
One person in KFC on that will lower.
Okay, thank you very much for muting that individual. Thank you. Just a reminder, keep your microphones muted at all times, guys. Thank you very much. Yeah, so the carbon monoxide inhibits the NADPH oxidases, reduces reactive oxygen species production, so prevents excessive damage to lungs. We also got iron being produced through the action of heme oxygenase 1 degrading heme. So heme is degraded by heme oxygenase 1 producing iron. Iron inhibits the replication of viruses, and also the production of biliverdin, as I said, due to breakdown product of heme. Biliverdin actually prevents the maturation of viral particles as well. So this heme oxygenase 1, this enzyme here, which is regulated by Nrf2, switched on by Nrf2, having all these antiviral effects. In addition to that, it can also bind to interferon regulated factor 3, which goes into the cell, binds to interferon sensitive response element within DNA cells and fires off type 1 interferons. Now, type 1 interferons are obviously part of the cytokine storm. The problem is, and this is quite a common thing with Nrf2, it's a balance, and it's a delicate balance between stimulation, having too much of it, and not having enough. So the cytokine, in the cytokine storm, we actually want to prevent too much interferons being produced, too much cytokines being produced, too much interleukins being produced. But we also need some to be produced to have the natural antiviral effects. So there's a balancing act to be got, to be had between too much and too little. Further actions of Nrf2, so not only does it induce lots of genes and heme oxygenase 1, we just saw on the previous slide, it also induces things like glutathione. Glutathione is a really important antioxidant which prevents lots of cellular damage. So the induction of glutathione is an important thing, prevent that damage to the cellular structures through that cytokine storm. And also Nrf2 induces other antioxidant molecules like thyrodoxin, pyridoxin, and thyrodoxin reductase. And these are all molecules that have an antioxidant, which act against oxidative stress and therefore are beneficial with regards to cytokine storm, reducing that damage. So we take all this into account then. It's proposed that Nrf2 activation will suppress that cytokine storm. It will reduce oxidative stress in the lungs, therefore reduce damage, and also might even aid in tissue repair, because there are actually some tissue-specific genes induced by Nrf2 in macrophages which are involved in tissue repair. So activation Nrf2 seems like a good idea. So obviously, as I said, we are interested in trying to find pharmacological activators of Nrf2, predominantly from a natural product source, to inhibit the replication of SARS-CoV2, or indeed both, or dampen the inflammatory response. Now, either inhibit that replication or dampen that inflammatory response using natural product activators of Nrf2. Now, interesting that DDEM in the last slides talked about licorice. I'm going to talk about licorice as well, because licorice, the plant Glycyrrhiza glabra, was actually found to inhibit the replication of SARS. So this is not SARS-CoV2, this is not COVID-19, but in 2003, when the SARS virus was emerged in Asia, licorice was actually found to inhibit replication of SARS in clinical isolates from patients with that disease. So that was a good indication that licorice would have an antiviral effect, inhibiting that replication of that SARS virus. However, the role of Nrf2 was never investigated with regards to whether Nrf2 was involved in the inhibition of that replication of that SARS virus. But coincidentally, within the CNPD, we actually did some work in 2016. This work was actually done by Naraza Bazar, Dr. Naraza Bazar, actually, as a postdoc when she was in the CNPD in 2016. And so actually, Naraza is actually talking tomorrow. I don't know if she's got technical difficulties. Hope she'll manage to come along tomorrow and talk. But she did some work when she was a postdoc with us, and she actually looked at the ability of licorice, Glycyrrhiza glabra, to actually induce Nrf2. So this is luciferase activity on the left-hand side. You can read that as being Nrf2 activation. And you can see that it's quite clearly seen that different fractions of licorice activated Nrf2 actually more than the control. Here's the control. Control is at a value of 1. We've got TBHQ, which is a positive control, at about 1.7. And we've got some other fractions, certainly fraction P20, maybe fraction P12, actually activating Nrf2 more than the positive control. So that potentially
Dr. Ritchie, you have some 2 minutes left.
Yeah, thank you very much. Thank you. So there is evidence, there is a suggestion here then that this antiviral effect of licorice actually may be due to the activation of Nrf2 by licorice. So that's an interesting thing we can maybe potentially pursue in the future. It'd be remiss of me to talk about Nrf2 without mentioning sulforaphane. The sulforaphane is a naturally occurring isothiocyanate found in cruciferous vegetables such as broccoli, and it's been shown to reduce virus-induced inflammation. So sulforaphane treatment of wild-type mice, reducing viral inflammation. Interestingly, that does not work in Nrf2 null mice, so mice which are devoid of Nrf2. Sulforaphane does not have that effect, suggesting that Nrf2 is driving that anti-inflammatory, anti-viral effect. We also see reduced viral entry and replication of influenza A infection following sulforaphane treatment, and also we see lower levels of interleukin-6 and viral load following treatment with sulforaphane in volunteers who have been infected with an inactive attenuated virus. So all this evidence suggests that Nrf2 has a significant antiviral effect, and that it is it certainly has a role to play in the prevention of infection and the detrimental effects of SARS-CoV-2 infection. So as you can see in conclusion then, we really need to find some more natural products that activate Nrf2, not only from the cancer point of view of preventing cancer, but also from the COVID-19 point of view to suppress viral infection, suppress inflammation, and hopefully prevent lung damage and aid the treatment and aid the recovery of patients. Obviously, with thanks to everyone in the CNPD for their support and help, Professor Sarkar and the rest of the CNPD, and also special thanks to Dr. Bazar, who actually did this work a couple of years ago, looking at effects of licorice being able to activate Nrf2. Thank you very much.
Thank you very much, Kenny. I'll remind the participants that there's a room to ask questions at the end of the session, so after the next speaker. But also, if you have questions, if questions bubble up during the talks,